We are continuing with the topic human health and disease. It's the lecture number 4, and today we will discuss about CMI, that is cell mediated immunity. See, first of all, I'll discuss about cells which are having a role in acquired immunity. There are two types of cells which play a role in acquired immunity: lymphocytes and antigen presenting cells, APCs. Now what antigen presenting cells do, they will recognize the antigen, process it, and then would present those antigens to lymphocytes so that lymphocytes can act over them. Basically, there are two types of APCs: macrophages and dendritic cells. If we talk about lymphocytes, lymphocytes are called as master of immune system because they are having a complete control on the immune system. They stimulate immune system, they suppress immune system, and even they are the effector cells of the immune system. In our body, there are around 1 trillion lymphocytes present. A trillion means 10 raised to the power 12. Lymphocytes are of basically three types: T lymphocytes, T cells or thymocytes, B lymphocytes or B cells, and natural killer cells or NK cells. We discussed about natural killer cells in our previous videos. What are T cells? T cells are produced and then mature in thymus, produced in bone marrow, and then they mature in thymus. Around 75 to 80% of lymphocytes are T lymphocytes, 10 to 15% of them are B lymphocytes, and 5 to 15% of them are NK cells or natural killer cells. And T cells are responsible for cell mediated immunity. B lymphocytes, they are produced in bone marrow as well as they mature in bone marrow, and they are responsible for humoral immunity or antibody mediated immunity. We'll be discussing about CMI in this video. That's why I taught you about the lymphocytes. Now the next category is natural killer cells, which are large and granular cells. See, B lymphocytes and T lymphocytes are agranular. That means there are no granules present in T and B cells. But natural killer cells is a type of lymphocyte, but it is having granules in the cytoplasm, and plus it's having a role in innate immunity. It has got no role in acquired immunity. T cells and B cells perform acquired immunity, but natural killer cells is having a role in innate immunity, and it kills virus infected cells and tumor cells. Rest all we discussed in our previous video, so we won't be having a good discussion on NK cells in this video. Now, as we know now, cell mediated immunity is performed by T cells. Now I'll talk about types of T cells which are present in our body. There are basically two categories in which I can divide T cells. The one is the effector T cell and the second is regulator T cells. Effector T cells are the cells which produce an effect for immune system. That means they are the actual T cells which will kill the virus, which will kill the infected cells. And regulator T cells regulate immunity. That means they can inhibit immunity or they can stimulate immunity. Activation of immunity or inhibition of immunity is done by regulator T cell, and the main immunity is provided by effector T cell. That means the direct effect is produced by effector cells, hence the name. Effector T cells are of two types: cytotoxic T cells and memory T cells. Cyto means cells and toxic means lethal. That means T cells are those those T cells which are having a toxic effect on the infected cells. That means they kill infected cells and tumor cells. Memory cells, now as we know, as we have discussed in our previous previous lectures, memory T cells are responsible for the secondary immune response, and whenever your body is having an encounter with the same antigen or pathogen the second or the third time, then your memory T cells transforms into cytotoxic T cells and having a rapid action against those antigens or pathogens. Regulator T cells are of two types. First one is the helper T cells, TH. As the name suggests, it helps in the stimulation of immunity. That means it activates the immunity. It activates both TC as well as B cells. When TC is activated, it would kill infected cells and tumor cells, and when B cells are activated, they would stimulate them to form antibodies, which is responsible for antibody mediated immunity. Now if I talk about suppressor T cell, what does suppressor mean? Suppressor means to reduce, to suppress. That means suppressor T cells suppresses your immunity by inhibiting TC and B. That means it inhibits both B cells as well as T cells. And why would your suppressor T cells inhibit your immunity? Only when the infection has been controlled. I'll be having a good discussion over it in a few minutes. I'm having some good questions for you here. The first question is, which type of immunity is activated by helper T cells? These are 4 options. The second question is, which type of immunity is suppressed by suppressor T cells? And both options are same for this. See, most of the students, what they do, they just see the word T cells and they tick the answer first, that is cell mediated immunity. They think that T cells are responsible for CMI, so the answer would be CMI, but it is not like this. As we discussed just a minute before, that helper T cells activates TC as well as B cells, and B cells are responsible for AMI, and also suppressor T cells inhibit TC as well as your B cells, which is responsible for AMI. So our answer would be both. That means helper T cells activates both CMI and AMI, and suppressor T cells suppress both CMI and AMI. Next question is, which type of cell can prevent autoimmune disease? Now, what are autoimmune disease? Autoimmune disease are the disease in which your immunity is having an interaction with the self antigens of the body. Whenever your immunity is producing antibodies against your own antigens, then that would cause autoimmune disease. So which type of cells can prevent this? So the answer would be T suppressor cells, because it helps in the suppression of immune system, and that is necessary. See, if the infection has already been controlled, and if your body would be continually producing antibodies or other immune products, then that may affect your own body cells producing autoimmune disorders. Now I'll be having a discussion on cluster of differentiation molecules, that is CD molecules. What are CD molecules? These are some protein molecules which are present on the surface of T cells and B cells, and around 250 different kinds of CD molecules have been identified till date. Now I'm discussing about helper T cells. See, helper T cells, on the surface of the helper T cells, some CD4 molecules are present. CD molecules are of different types and they are named as per the sequence CD1, CD2, CD3, CD4 and so on. Helper T cells have got CD4 molecules on their surface, and hence they are called as CD4 plus cells. And suppressor T cells, the TS cells, are having CD8 molecules on surface, and hence they are called as CD8 plus cells. Now, how to remember which one is CD4 and which one is CD8? I'll write H of helper, only half H. This is half H. Now I can convert it into 4. That means helper T cell is CD4. Write 8. This is S, and it can be easily transformed into 8. That means, in short, S resembles 8 and H resembles 4. By this way, you can remember that helper T cells are CD4 plus cells and suppressor T cells are CD8 plus cells. And the most important question is the ratio of CD4 is to CD8 in the body, and the answer is 2:1. So you need to remember the normal ratio. Now, a very important topic, that is activation of acquired immunity. How acquired immunity gets activated in the body? I'll start with an antigen entering our body. Whenever any antigen enters the body, first of all, APCs, antigen presenting cells, would process, recognize and present these cells to the lymphocytes, and the examples are dendritic cells and macrophages, like we discussed earlier. APCs would present antigens to the T helper cells, and now T helper cells get activated by interacting with that antigen. That means antigen is brought first to T helper cell. T helper cell interacts with the antigen and gets activated, and when T helper cells gets activated, it leads to activation of B cells and TC cells. When B cells are activated, it forms antibodies against that antigen, and when TC, cytotoxic T cells, are activated, they would directly target the infected cells. That means the first cells of immunity, the first lymphocytes which gets activated are the T helper cell, and this is a very important question. Now I'll be having a small discussion about HIV, the virus that causes AIDS. HIV's prime target is T helper cell. Whenever HIV enters your body, it targets T helper cells, and slowly the number of T helper cells starts decreasing in the body. And now we can very much relate this. Whenever there would be a deficiency of T helper cells, cells, your activation of immunity is hampered, and that leads to deficiency of immune system. That means immunodeficiency, and that's why HIV causes immunodeficiency. So after target infected cells and antibodies are produced, the infection is now under control, and when the infection is under control, T suppressor cells are activated. They would now inhibit B cells and T C cells because the work has been done. Now a very important topic, clonal selection of lymphocytes. Now to understand this topic, I'm having a small question for you. See, whenever any antigen enters your body, how come in a very short time your body produces enormous number of lymphocytes, enormous number of antibodies against that same antigen? There are plethora of antigens in this universe. A single bacterial surface can have multiple different agents. So your body cannot have cannot already have thousands of already formed specific lymphocytes or specific antibodies against any antigen, because we know antigens and antibodies are very specific, and acquired immunity, as we know, is very specific. So how is it possible? So the answer to that is clonal selection of lymphocyte. Now listen to this carefully. I'll start with an antigen, a small blue triangle entering our body, and when antigen enters, there are some receptors present on the surface of lymphocytes which interact with those antigens. Now the receptors that are present on the surface of lymphocytes are very specific towards this antigen. Let's say an antigen has entered in our body, and these are many lymphocytes present in our system, but out of these many antigens, few of them has got some receptors which would interact with this antigen because these are very specific. So those lymphocytes are selected whose receptors are specific to interact with the antigen. As you can see in this figure, there is a green lymphocyte with the red receptor which is specific to this blue antigen. So this blue antigen now would come in contact with the receptor, and both of them are very specific. Now what would happen? Your lymphocytes will get activated and they will start dividing to form multiple clones. What are clones? Clones means exact Xerox, that means copy of cells. So these lymphocytes with this specific receptor against that specific antigen would produce multiple clones of itself, and multiple number of cells are produced with the same receptor that would act against that antigen. Now these lymphocytes would transform into different types of effector cells, but even after transformation, the receptor on them would be specific to that antigen. Now what type of effector cells are transformed? The first one of them are the B cells. Like this, the lymphocytes have now transformed into B cell, but the receptor is still the same. As I have shown you in this figure, the red receptor is always same, which is specific to this blue antigen. This is just for an example for you. Now when such B cells are transformed, then they would produce antibodies, and these antibodies would fight against these antigens. And the second transformation occurs in the form of T cytotoxic cells, again with the same receptor that would fight against that same antigen. And the third one are the memory cells that would be transformed, and same receptor would be present even on their surface to fight against those antigens. B cells, they would produce specific antibodies that would fight against the antigen. Cytotoxic T cells would directly target the infected cells, and memory cells would be present in your body for a long time, and whenever the same antigen would enter your body for the second or the third or any consecutive time, then these memory cells with the same receptor that is present in them would be converting into TC cells and would produce a heightened response called as secondary immune response. So that is the meaning of clonal selection of lymphocytes. Thank you very much for watching us. Keep connected with us so that we can go for our next lecture, which would be on the humoral immunity or antibody mediated immunity. Thank you very much for watching us.